Our last talk for the CCR this year is, again, Mike McClung. He's going to talk about treatments in development for osteoporosis.
We've already sort of set the stage for why there is an opportunity, or why there are opportunities for new drug developments in osteoporosis. And so let me share with you some of the information and action that's happening in that field. Again, disclosures before, so all of the drugs that I'm going to mention in development I have worked with or have consulted about as well. So the treatment benefits we've talked about, the limitations for therapy we've already talked about, the two major doors that are open for new drug development are, number one, our drugs, although they're beneficial and effective in reducing fracture risk, they don't cure osteoporosis. And particularly, there's a deficit in our capability of reducing the risk of non-spine fracture. None of our drugs do a very good job of reducing the risk of non-spine, especially non-spine, not non-hip fracture. And then the other is that the concerns, again, about the safety and tolerability that we've already beat to death this morning. So let me at least share with you that there are new drugs in development in both the anti-resorptive and the anabolic fields. There are new forms of old friends, and then there are some totally new strategies based on new findings and new approaches that have to do with the molecular biology of bone. So I'll deal with some, but not all of these things. Raloxifene, we've already talked about as a selective estrogen receptor modulator. The search for the ideal SERM was undertaken by many companies a number of years ago, and several more SERMs that were shown to be more potent than raloxifene in preclinical studies and even in some clinical studies underwent evaluation. And that includes lasofoxifene, arzoxifene, and basodoxifene. I'll mention only one of these. I'll show you the data in a moment about lasofoxifene. Arzoxifene was also developed by Lilly, who makes raloxifene, and in studies that we and others did, arzoxifene resulted in a modestly greater increase in bone mineral density, a modestly greater decrease in indices of bone turnover, yet in the large clinical trial that was done and recently published by Steve Cummings and others of us, the effect of arzoxifene on reducing fracture risk and its side effect profile was indistinguishable from raloxifene, and so its development was curtailed. Basodoxifene similarly actually did a head-to-head study, including against placebo and raloxifene, with fracture outcomes with basodoxifene being identical to raloxifene. And so its evaluation as an osteoporosis drug has essentially been halted. The company is still toying with the idea of developing and using basodoxifene in combination with estrogen. And in that sense, basodoxifene would be used not because of its effect on the skeleton, but because it would inhibit the action of estrogen in the endometrium. And so the combination of estrogen plus basodoxifene would be used in women who have not had a hysterectomy in lieu of using a progestin to protect women from the endometrial effects of long-term estrogen therapy. Lasofoxifene is approved for the treatment of osteoporosis in Europe and in other parts of the world. It will likely not be submitted for approval in the United States. I've already talked about some of those things. Here are some data from lasofoxifene. We, again, compared lasofoxifene with raloxifene and showed that it had a better improvement in bone mineral density than did raloxifene in postmenopausal women without osteoporosis, and more potently inhibited markers of bone resorption. So the company was enthusiastic that this perhaps was going to be a more potent and better therapy. In the large phase 3 study that was published last year by Steve Cummings, the effect of lasofoxifene on both spine and non-vertebral fracture was shown to be statistically significant. So 2 doses of lasofoxifene were studied, and the higher dose of lasofoxifene reduced the incidence of new spine fracture by 42%, and for the first time, a non-bisphosphonate, non-denosumab anti-resorptive drug was actually shown to reduce the risk of non-vertebral fracture with a reduction of non-vertebral fracture of a little more than 20%. So this suggests, at least, that lasofoxifene might be more effective than raloxifene. Again, not a head-to-head study. It's not quite fair to compare results across studies. The fact that we haven't demonstrated non-vertebral fracture risk reduction with raloxifene doesn't mean it doesn't work if we had done a bigger, better study. But at least for the first time, we have evidence that a SERM can reduce the incidence of non-vertebral fracture to the same extent that we see with other potent antiresorptive drugs. The other outcomes that were observed were what we normally see with SERMs: a clear reduction in the incidence of estrogen receptor positive invasive breast cancer, an estrogen-like increase in the incidence of venous thrombotic events, no increased risk of stroke or heart disease was observed. So lasofoxifene might be a better version of a SERM, but because of some other safety concerns, predominantly endometrial safety, there has been reluctance on the part of the sponsor of the study to submit these data for review by the FDA. So whether we'll see a new SERM or not is unclear. Even if we do, SERMs are not the first-line strategy for most of our patients with osteoporosis anyway. Calcitonin is probably the perfect antiresorptive drug. It's the natural inhibitor of osteoclast. There haven't been any safety concerns. I didn't know either about the lithium story of calcitonin. But the nasal formulation of calcitonin has been difficult to demonstrate any effectiveness, and most of us think that the data suggesting that nasal calcitonin has important benefits is very sparse. Oral formulations of calcitonin are being evaluated, and at least in some early studies, there is a suggestion that calcitonin administered in a new oral formulation has the capability of significantly reducing biochemical indices of bone turnover. Interestingly, this is a very short-term effect, as seen here 3 hours after the dosing of oral salmon calcitonin. There's a clear reduction in the level of CTX, but that effect is gone by 24 hours after the dose. A phase 3 study evaluating the effect of this oral salmon calcitonin preparation is just being concluded. It's administered once a day. It will be interesting to see if transient inhibition of bone resorption gets translated into fracture risk assessment. So it may be that calcitonin will sort of re-arise or re-emerge in a new form and be an interesting option for patients with osteoporosis. The effects, of course, of our therapies, as we've talked about before, are really based on our understanding of bone remodeling and are targeted either to reduce osteoclastic activity or to increase osteoblastic bone formation, or maybe ideally a combination of those two things. We've already talked about that we have understood or are understanding better the molecular signals by which bone metabolism is regulated and by which the crosstalk among the various bone cells actually occurs. And some of the new strategies are based upon those new observations and new understandings. So let me talk about 2 other new drugs that are in development that actually I think are very exciting and have the potential for actually being more effective than our current drugs. The first of these are drugs that inhibit cathepsin K. Cathepsin K is a member of the cysteine protease family of enzymes whose job is to digest collagen. Some of the cathepsin molecules are ubiquitously distributed and through all kinds of tissues to keep collagen levels intact and under control. Cathepsin K is relatively specific